Hello friends, welcome back to a new video, that is MCQs on cell signaling, and this is the part 3 of this series of videos on cell signaling, and also it is the last part of this cell signaling video. So the first question of this video, that is one of the major causes of Plasmodium falciparum resistance to chloroquine is amplification of the gene for option A MSBA transporter gene, option B Lassie permease gene, or option C AQPZ porine gene, or option D ABC transporter gene. So one of the major cause of Plasmodium falciparum resistance to chloroquine is amplification of the gene for option D, that is ABC transporter. So ABC transporter is responsible for resistance of Plasmodium falciparum to chloroquine antibiotic. So this, what is this ABC transporter? This ABC transporter, which is ATP binding cascade transporter, are a superfamily of membrane protein. This is a membrane protein that move substances across membranes. So they are found in all prokaryotes, plants, fungi, yeast and animals. So how they helps the Plasmodium falciparum to resistant to their chloroquine. So because this ABC transporter is increased in their site, in their membrane, so that's why this when we apply this chloroquine, they get out from this, from their cell membrane, from this from their inside the cell, so they cannot enter into the cell. That is the reason of resistance to chloroquine. They get out from this membrane, their cytoplasm, to the outer side. Okay, so this is the first question of this video. So let's move on to question number 2, that is SH2 domains are option A protein domains that bind phosphotyrosine containing peptides, or option B the domains on receptor protein tyrosine kinase that contain the phosphorylated tyrosine, or option C that this SH2 domains that mediate the dimerization of receptor protein tyrosine kinase, or option D the domains on receptor protein tyrosine kinase that possess the kinase activity. So which one is true about the SH2 domain? So the right answer is option A, that is these SH2 domains are protein domains that bind phosphotyrosine containing peptides. So here we will see this SH2 or SRC homology 2 domains are protein molecules that bind to phosphorylated tyrosine residues on other proteins, and they are found in many proteins involved in tyrosine kinase signaling cascades. Okay, so next question क्या है? Next is rhodopsin and muscarinic acetylcholine receptors share which of the following features? And the options are option A, each causes an inhibitory intracellular response, option B is each activates a tyrosine kinase cascade, option C each is composed of an alpha beta dimer, or option D each functions through a heterotrimeric G protein. So which one is correct about this rhodopsin and acetylcholine? So the right answer is option D. This rhodopsin and acetylcholine receptors share the function, that is each functions through a heterotrimeric G protein. Okay, so the next question is which of the following statement about the actions or targets of the second messengers of the phosphoinositide cascade is incorrect? And the options are option A DAG activates protein kinase C, that is PKC, protein kinase C. DAG activates protein kinase C, or option B most of the effects of IP3 and DAG are antagonistic. Option C DAG increases the affinity of PKC for calcium, or option D PKC requires calcium for its activity. So the right answer is option B, that is most of the effects of IP3 and DAG are antagonistic. So here we will see the phosphoinositide pathway is known to involve receptor stimulation of phospholipase C, that is PLC, via GQ or GI type G protein to stimulate the formation of inositol triphosphate, that is IP3, and diacyl glycerol DAG as second messengers. Okay, so this so this action or target of the second messenger of the phosphoinositide cascade, which is incorrect. The most of the effect of IP3 and DAG are antagonistic. This is incorrect. Okay, so let's move on to question number 45. It is also same. So which of the following are the second messenger that are produced by the phosphoinositide cascade? And the options are option P, that is phosphatidyl inositol 4,5 bisphosphate, that is PIP2, and option Q, that is inositol 1,4,5 triphosphate, option R, that is inositol 4-phosphate, option S, that is diacylglycerol, it is DAG. So by this phosphoinositide cascade, which one is the which one is the secondary messenger produced by this phosphoinositide cascade? And options are P and Q is the right one, option B R and S is the right one, option C Q and S is right one, or option D P and R is the right one. So the right answer is option C, that is Q and S is the right answer. So Q, that is inositol triphosphate, and S, that is diacylglycerol. These are the two secondary messenger which are produced from phosphoinositide cascade. Okay, so let's move on to question number 46. That is, the MEK kinase is unusual in that option A, it is activated by a kinase. Option B, it lies downstream of G protein coupled receptors. Option C, it is a dual specificity kinase having the ability to phosphorylate both threonines and tyrosines. Option D, it activates a kinase. So which one is the right one? That is option C. That is, this MEK kinase is unusual in that it is a dual specificity kinase having the ability to phosphorylate both threonines and tyrosines. Okay, and what is this MEK kinase? This MEK means mitogen activated protein kinase. It is a kinase enzyme that phosphorylates mitogen activated protein kinases, that is MAP kinases. It activates this MEK activates the MAP kinases, and this MEK is also known as MAP2K and MAPKK. So MEK phosphorylates threonine and tyrosine residues to activate extracellular signal regulated kinases. Okay, so let's move on to question number 47. That is, a signal sequence KDEL is removed from ear resident protein. So assuming that there is no change in tertiary structure of such plant protein and no other signal sequences present in protein, the changed protein will now have following fate. So options are option A, it will remain in ear and be degraded. Option B, it will be targeted to Golgi apparatus. Option C, it will be secreted outside the cell, and option D, it will be targeted to lysosome for degradation. So the question is, a signal sequence, that is KDEL, which is present in a protein, signal sequence which is present in a protein, ER resident protein, but this KDEL is removed. It is removed from that protein. So what will be the consequence? Okay, so the right option for this situation is option C, that is it will be secreted outside the cell. So if we remove the KDL sequence or signal sequence from this ER resident protein, they will be secreted outside the cell. So what is KDL? So KDEL is a target peptide sequence in mammals and plants located on the C-terminal end of the amino acid structure of a protein. So the KDEL sequence prevents a protein from being secreted from the endoplasmic reticulum and facilitates its return if it is accidentally exported. So if KDEL sequence is removed from the protein, then they will be secreted from the ER. Okay. So next is question number 48. So which of the following is true regarding influenza viruses? And the options are: option A, mutations are responsible for pandemics; option B, no effective vaccine is available; option C, HA protein is responsible for release of virus particles from infected cell; or option D, genome has 8 segments. So which one is correct about influenza virus? The correct one is option D, that is genome has 8 segments. So this influenza virus has genomes has 8 segments. Okay, so the influenza virus genome is made up of 8 single stranded RNA segments. These segments are negative sense molecules, which means they must be copied into positive sense molecules to produce proteins. Okay, so the last question of this video, that is CHEA, this is a protein that participates in bacterial chemotaxis, and the statements about this bacterial chemotaxis and this CHEA protein is this statement P is the CHEA protein is a histidine kinase, and the next one is it shows autophosphorylation, and the next statement R is it transfers phosphoryl group to conserved aspartate residues in the CHEY, and the statement S is it is a tyrosine kinase. So which option about this CHEA protein is correct? So the option A is P and Q is correct, option B R and S is correct, option C P Q R is correct, or option D P Q R S. All options are correct. So the right answer is option C, that is PQR is the correct one. So the CHE protein is histidine kinase, and it shows autophosphorylation, and it transfers phosphoryl group to conserved aspartate residue in the CHEY. So this is the correct one. So this CHEA is a histidine kinase protein that regulates bacterial chemotaxis. It is a part of a family of protein that control gene expression in response to environmental changes. Okay. So the last question of this video, that is necrosis that develops in tissues subsequent to denaturation of structural and enzymatic proteins soon after death is appropriately referred to as: option A, fat necrosis; option B, liquefactive necrosis; option C, coagulative necrosis; or option D, caseous necrosis. And the right one is option C, that is coagulative necrosis. So necrosis that develop in tissues subsequent to denaturation of structural and enzymatic proteins soon after death is appropriately referred to as coagulative necrosis. Okay, so these are all questions on cell signaling. All this, and this is the part 3 of this cell signaling video. Okay, so thank you for watching this video.